Now we're going to move on to our next laureate, who is Dr. Ronald Vail. He joins us from the University of California, San Francisco, where he is professor of cellular molecular pharmacology, so we have another pharmacology department joining in the group. He's also a Howard Hughes medical investigator, and as many of you probably know, next year he's taking on the leadership of the Genelia Research Campus at Howard Hughes. And there's a connection with the previous talk, as you'll hear. Dr. Vail was awarded for the landmark discovery of the motor protein kinesin and pioneering the understanding of its mechanism of action in driving cellular movement processes, including cell division and intracellular transport. And his lecture is called How Living Organisms Generate Motion.
It's a great pleasure to be here, an enormous pleasure to receive this very prestigious award, and it's also wonderful to follow Susan. I'm not sure if we met before, but it was great to hear about your pioneering work, and actually Susan's discovery of Taxol really made all of the work that I'm going to describe possible when I get into telling you about the biochemistry of kinesin. So I'm also going to frame this talk at a very general level. I've heard there are also non-scientists in the audience as well. But I'm going to tell you about a class of proteins called molecular motors and our work on different kinds of molecular motors called kinesins and dyneins. So first of all, biological motion is something that everyone is familiar with. We're all familiar with the fact that we walked into this auditorium, and that involves muscles. Even preschool children are aware that the biological world is full of life. And one of the obvious things when you look under a microscope and take any pond water and look at it, you see, as Leeuwenhoek first did, that it's also teeming with movement. Various types of single-cell organisms that are swimming around using cilia and flagella. And now if you peer in the microscope at even higher magnification and look inside of cells, again, the most obvious feature is that it's packed with different types of motility. This is what I'll tell you about later, movement inside of nerve cells, but you can see all kinds of objects here. Small membrane organelles and long mitochondria that are moving inside of the interior of cells. And indeed, if you look at virtually any cell in your body under a microscope, you'll see that it's full of objects moving inside of the cell. And you'll hear later about DNA replication, which produces replicated chromosomes, which then have to divide. And these are the chromosomes here in green. And you can see in the process of mitosis that they physically separate from one another. And that's, again, a kind of biological motility that involves these molecular machines that I'll talk to you about. Really, all of this motility that I've been describing to you has analogies to the way we generate motility in our cities. It involves roadways. And you heard beautifully from Susan's talk about a very important roadway called microtubules. And on these roadways, there are machines that move along those roadways, often much more efficiently than one will experience in Toronto. Taking the analogy to this movie, goods have to be shipped from one place in the cell to the other. Those goods are in these cargo containers that have to be moved by motors. And if we look inside of cells, we find analogous systems. So one are the microtubules that you heard about from Susan. Those are these fibers that you see here. And on these fibers are packages of material which are contained in these membrane vesicles. And these membrane vesicles are moving along, that's the microtubule that you heard about from the last talk, are moving along these microtubule tracks. And it's these machines that I'd like to tell you more about. So in the human body, there are about 100 different types of molecular motors, protein machines that move along tracks. And one group of track are actin filaments, and there are about 35 different types of motors called myosins that move along those tracks. Most notably, the myosins are the machines that cause your muscle to contract and your heart to beat. And along the microtubules that you heard about from Susan, there are two classes of molecular motors, kinesins and dyneins. And within each of those, there are many different subtypes. So there are about 100 different types of motors moving along tracks. And the reason why there's so many is that these motors are specialized for all types of different kinds of motility. Some are involved in cell division. Others are involved in transporting these membrane vesicles inside of nerve cells. Other ones are transporting RNAs. So given the whole complexity of the different types of movement that have to occur, there have become specialized motors that are dedicated to different tasks. So this is what this talk is about, but maybe I can even preface this by first saying, why do we study them? Why are they interesting? Well, as I mentioned before, everywhere you look, movement is an essential part of life. And this even starts at development, from the process of fertilization to the development of an embryo to the development of a full human being. Motors are involved in moving materials such as RNAs and proteins inside of cells that generate a lot of the fundamental principles of embryogenesis. I showed you already that cell division here, another fundamental aspect of life, involves these molecular machines. The other reason is that these motor proteins are fundamentally fascinating machines in how they work, as I hope to show you. But I think they've also provided a paradigm for what I consider a big challenge in biology, which is to understand the protein machines inside of the cell. We're obviously living in an omics revolution where we can measure, all the genes at once. But we have to remember that those genes are making something at the end of the day. They're making protein machines that are allowing the cell to work. And the better we understand these machines, the better we're going to understand biology, whether it's a motor protein or Cas9, another amazing machine here that was celebrated with the Gairdner Prize a few years ago with Rodolphe in the audience here. But these are examples where understanding these machines leads to very important practical outcomes and also an important understanding of biology. And some of the practical outcomes do, in addition to being fascinating, there are practical outcomes out of this. So first of all, in motility, there are a number of different genetic diseases that involve motors or motor-associated proteins. And when things go wrong, that can lead to neurodegenerative diseases, infertility, and a whole variety of cardiac misfunctions as well. But there are also opportunities that if we understand these motors, just like you heard with Taxol and the roadways, these motors can be manipulated by small molecule drugs. And just one example of this is a drug that targets the myosin motor that causes your heart to contract. And this was an effort that was undertaken by a company called Cytokinetics that I was involved in co-founding along with James Sabry, who may be here somewhere in the audience, and Larry Goldstein and Jim Spudich, to really take our understanding of motor proteins and develop drugs against them that can do potentially useful things for therapy. And this is one amazing drug called omecamtiv, a program that was developed by actually my very first graduate student in the lab, who became the first employee at this company and is now, led this amazing drug discovery effort. But it was a very audacious project to try to make this motor actually work better than it does naturally, to activate the activity of this motor protein. And why would you want to do that? Well, there are some patients where the motor is not fully contracting to cause the heart to contract properly, and that gives rise to failure. So there are many people with heart failure that the contractility of the heart is not working normally. And this drug actually improves the performance of the myosin and improves the contractility of the heart. And this just shows an example of a patient with heart failure on the left. You can see the mitral valve here kind of fluttering, and that's because the heart is not contracting properly. But several hours after taking this drug, you can now see this mitral valve popping open, and that's because the drug has activated the molecular motor and is causing the heart to contract more vigorously. So now this drug, after many years, is now in an 8,000-person clinical trial around the world to see if this will improve outcomes of patients in terms of morbidity and mortality with heart failure. But what I would say is that this program required a detailed biochemical and biophysical understanding of the myosin machine at the onset. Otherwise, this whole drug discovery program would not have been possible. So this leads me into next part, which is to tell you a little bit about now how these motors work. And I'll maybe end my talk, if I have enough time, just to tell you about another passion that I have, which is about science communication, which I know is also very fundamental to the mission of the Gairdner Foundation as well. Okay, so I've had like two eras of work on molecular motors. One era, I suppose the kinesin era, occurred over about a couple decades, and that happened when I was a graduate student in Eric Shooter's lab at Stanford, and work in his lab got me thinking about the general problem that eventually led to the discovery of kinesin. And the discovery of kinesin occurred at the Marine Biological Laboratory at Woods Hole, Massachusetts, with this group, Mike Sheets, Tom Rees, Bruce Schnapp, and myself, looking a little bit younger. I just had a mustache, that's the only difference. But it was this collaboration that was involved in the work that I'll briefly tell you about. So the problem that I got interested in from my work in Eric Shooter was how material is transported inside of nerve cells. So nerve cells are interesting because indeed they are microscopic in size, but they're extraordinarily long cells. They're the longest cells in your body because one part of that cell is where your DNA and nucleus is. That's called the cell body. And that cell body may reside in your spinal cord. But then there's a long process that extends from that cell body, which is called the axon. And that's the part that's found in your nerve, which is a combination of many, many axons. And at the very end, which could be at the tip of your toe, is the nerve terminal, which may be controlling the movement of a muscle, for example. And the axon conducts an electrical signal to the nerve terminal to control the muscle. But also, there's another kind of transport that occurs in this axon, which is the shipping of goods between the cell body and the nerve terminal. So essentially, most of the proteins that are made and lipids that are made in the cell body, but they have to be shipped to the nerve terminal, just like shipping goods in a city. And also materials that are taken up at the nerve terminal have to be sent back to the cell body. So it's a bidirectional highway in the cell occurring over a distance of about 1 meter, which is an extraordinary distance at a cellular level. And at the time we were working on this, the mechanism of how that transport worked was not really understood. So if one's going to tackle a problem in biology, it's always important to find a good experimental organism. And they can come from surprising places. And in this case, the experimental organism of choice was a squid. Why is that? The squid has an axon that's 50 to 100 times bigger in diameter than a human axon. That makes it possible to dissect out of the squid, get biochemical material to study this process. So it also had an important history, actually, in neurobiology, because the electrical conductance of the nervous system was figured out in the squid giant axon as well. So this is what the squid giant axon looks like if you look at it under a microscope, a video microscope, which at that time in the early 80s was a newly developed technology, putting a video camera on a microscope. That was developed by Robert Allen and Shinya Inoue, and Allen, working with Brady and Lassick, made these movies of the squid giant axon, which were incredible, because for the first time, you could peer into the secret world of the axon and see all this transport occurring. However, one didn't really know at that time what was the machinery involved, and it wasn't even certain that microtubules were involved in that process at the time. So the goal then was to take this complicated system and to begin to dissect it in a test tube environment, to essentially take out the interior of the squid giant axon and try to get this transport system to work in a test tube environment so you can begin to take it apart. And here was an experiment that I'm very grateful to Susan and her Taxol for. In fact, I either wrote to you or the NCI or someone who provided Taxol for free for these experiments. Now, what you're seeing, the difference between here and here is that this is a test tube. These little lines here are microtubules that were purified from bovine brain, as Susan described, and stabilized with Taxol. So they were pure tubulin. They weren't the microtubules from the squid. They were from a cow, stabilized with Taxol. And in this test tube environment, we figured out, actually by complete serendipity, that's a whole other story and lecture, but that the natural cargo of these membrane organelles could be substituted for small plastic beads. So what you're seeing here is not a biological substance. It's approximately a 1 micron size plastic bead. And we were able to trick the motility system to transporting these artificial cargo along these purified microtubules. And of course, that raised the question of what was doing the transport. And at that time, we had no idea. So we then had to take the system further apart to figure out which is the molecule that's producing the transport. And a way of thinking about this is to take a complicated system with many parts and then figure out which of these parts is the part that you actually need to make this car run. And many of these parts are superficial here. So if you can take the car apart and figure out the essential pieces, then you can begin to figure out how a car engine works. So this strategy was similar. Take apart a very complicated system, and a squid may have 20,000 different proteins. But now, in this test tube environment with these plastic beads, figure out which of these 20,000 proteins is a protein that's causing this motility that I just showed you in that diagram. And this was done by biochemistry and fractionation. So, this was kind of a classic method of separating proteins. But here you could see an end stage of that purification where there's a protein band that correlates with movement under a microscope. And I didn't think about it very much at the time, but biochemistry was mostly done with radioactivity and spectrophotometers and things like this. This was probably maybe the first use of a microscope as a tool for biochemistry. And that's been actually an important theme kind of my career ever since that, that this is a very powerful combination. And out of this came a new molecule. So it turned out to be a protein that no one had discovered before, and that was named kinesin. Again, I credit the name to my mother, who I called on the phone, and because I was posed with a dilemma of what to name this protein, and she and her Greek friend came up with the name kine, a root for movement, and hence kinesin. So at that time, you can imagine just looking at these movement under the microscope was utterly captivating. And it was almost taunting, a taunting challenge to figure out how this worked. But at that time, this was like 1984, 85, it seemed beyond the realm of possibility to understand how a machine that was probably 1/1,000,000th of an inch in size worked. We couldn't see the motor, we can only see what it was transporting. And how are we going to understand the details of how such a small machine worked? And honestly, I kind of thought that would be maybe not achievable in my lifetime when I first made that discovery. And this just shows how fast science is changing, and what was an impossible task becomes possible. And that's what makes science so exciting right now, because I would say between our lab and the whole field, we actually understand how this machine, 1/1,000,000th of an inch, actually works. And that was done by, again, going to reconstitute things in a test tube. And another development that came out after this was the realization that these plastic beads and other kinds of assays, the motility could be produced by a single protein molecule. And out of that emerged kind of a whole new type of biochemistry, which was single molecule biochemistry, which in a way emerged out of the motor protein field collectively. Again, the realization that you can use light microscopy for biochemistry, and in fact, you can use light microscopy not to study billions of molecules in a test tube, but study individual machines, one molecule at a time. And the other thing that was important for understanding these machines was getting their structure at the atomic level. And again, that was already known, but that became easier with X-ray crystallography and cryo-EM. Now, one advance after the bead here was to remove the cargo. We can't see the motor here at all. Was to remove the bead completely and replace it with a fluorescent dye. And now we can measure the machine directly by fluorescence. And this was done in a collaboration with Toshio Yanagita in Japan when I was on sabbatical. And now what you're seeing are individual dots, which are single protein machines moving along these tracks. And you can now use kind of computational techniques, which were developed and we did collaboratively with Paul Selvin, to follow these fluorescent tracks in great detail at the nanometer scale level. And now you can see that the motor is actually taking steps which correspond to the stepping stones that you saw in Susan's talk along the microtubule track. So we can now measure the dynamics of how this machine walks. And an important thing to know about this machine is that these are chemical engines. They're enzymes. They get their energy for movement from a chemical energy source, which is ATP. And what these machines do is they hydrolyze the ATP, they release the phosphate group, they release the ADP, then they release the whole nucleotide, and they start again. And during each one of these chemical cycles, the protein takes one step. So the goal right now was to say, what are the transitions in this chemical cycle that cause this machine to take a step along the track? And an important element of understanding actually these chemical transitions was going to atomic information. And that summarized a lot of work just to say that first involved X-ray crystallography with Robert Frederick and John Kuhl and Elena Sabline to get the first structure of kinesin. And out of that actually came an enormous surprise in the field that everyone thought that kinesin and the myosin that moves your muscle were completely unrelated to one another. And from this structure here, although I won't show you the details, we found that kinesin and myosin were in fact evolutionarily related. And we could begin to dissect some common themes now, not just of how kinesin worked, but comparing kinesin to myosin kind of allowed us to develop some universal principles of how both of these classes of machines work. And so from these static structures, we also use then dynamic information from single molecule technique called fluorescence resonance energy transfer to understand how this machine changes its shape during the ATPase cycle. And I just summarized an enormous amount of work here by showing you an animation of the kinesin molecule. It's based upon atomic resolution information, and the dynamics are developed from a lot of microscopy information, but it is a model. But when ATP comes in here, you could see this little arm zipper up along the core of the blue part of the enzyme. Again, the ADP is released, the ATP comes in, you'll see the zippering action, and that causes the partner head to move from a rear site to a forward site. So I'd like to say it's a little bit like a leapfrog action of these two motor engines that take turns along the back or front position along the track, or a little bit like a judo expert that uses an arm to flip an opponent from a rear site to a forward site. And so that's this arm here. That's the mechanical action of the kinesin that allows it to move methodically along the track. Okay, as Susan said, this is built upon really the work of so many people that I'm grateful to, mentors, students, collaborators, and other people in the field that led to this understanding of kinesin. We then began, after this began to get worked out, we thought we'd turn our attention to another machine. Kinesin moves from the spinal cord to the nerve terminal, but dynein is another motor that moves in the opposite direction. It also causes ciliary beating, so it's the motor that causes cilia and flagella to beat. And a whole other group of people were involved in that effort. Particularly, I want to mention my really good friend and colleague, Ian Gibbons, the discoverer of dynein, who was a great friend and got us into this whole field, and now postdocs who have great labs of their own working on dynein. We went to the same playbook of techniques. It wasn't easy because dynein is about 10 times larger than the kinesin molecule, so it posed much greater challenges. But at the end of the day, we collectively got the atomic structure of dynein, a lot of dynamic single molecule information. Again, this is really summarizing our work, but other labs in the field. And you can see the dynein works by a very different mechanism. It's evolutionarily unrelated to kinesin and myosin, and it's kind of a giant on stilts here. And the ATPase cycle, what it does is it thrusts this kind of stilt forward on the microtubule track so it can rebind and kind of latch on to take a step to move forward. So it's a very different kind of moving machine than kinesin, which just shows you the diversity of machines that nature can evolve to produce movement. Again, the whole journey, I am blessed with a wonderful group of young people. And I think one of the great things about being a principal investigator is just having so many fantastic young people come to the lab and see them prosper and go on to their own careers. I will say, I probably have 2 minutes. I do want to say that I also share, in addition to my passion for science, a passion for communicating it. And I think this is a very important part of the mission statement of the Gairdner Foundation. I am a total believer in this. I think it's our responsibility also as Gairdner Award winners and senior scientists to be part of that inspiration in paying it forward. And in that spirit, I've been very much involved in a lot of communication projects, which I must admit, I think are as creative as science itself, and I think can be as impactful. Developed a project called iBiology. My wife, Karen Dell, has helped me in that, and she's here too. But it produces videos of leading scientists giving talks. There are about 500 talks right now, freely available to scientists throughout the world, including many places that don't have access to leading scientists and seminar series. I've just launched, with a lot of helpers, including Rodolphe here, who has contributed to this project, a brand new kind of quote-unquote textbook, because it's mostly online, for college biology, with a goal of making access free, because textbooks cost too much for many students around the world, and also to expose students not just to facts and memorizing facts, but exposing them to all the great things we're hearing today, what scientific discovery means. This is the website. It just launched 2 weeks ago. It's called explorebiology.org. I encourage you to take a look at it, and feel free to contact me later. Again, another project that involved Rodolphe, but through two very talented filmmakers, Adam Bolt, Elliot Kirshner, we also kind of launched a new effort in iBiology to make documentary films. This is about CRISPR, the whole discovery of CRISPR, and its impact in modern biology, called Human Nature. It just finished playing in Canada, but it will be playing in other venues soon. So I encourage you to look at this when it comes out. And I'm also very involved in improving scientific publication. That's another form of communication, of how we communicate to one another as scientists. And I think that's also very important for scientists to take a leading role in thinking about how to improve scientific publication. So with that, it's been a tremendous week. I've not only enjoyed today and will enjoy today, but I've enjoyed the whole week of talking to high school students and other universities. And I think this is just a wonderful thing that the Gairdner Foundation does. So thank you very much.